A radiation-resistant bacteria may be the key to creating an environment on Mars that can sustain life. These bacteria have a unique adaptation to their genetic sequence that allows them to survive intense ionizing radiation. This allows for a backup to replace any part of the genetic code that is damaged by radiation. It has been proposed before that this microbe could possibly survive on Mars. Indeed it may, however it will be one of the only microbes that can survive. The goal is to create an environment that is hospitable for many different life forms. Synthetic biology is an up-and-coming field. A new technology of taking coded DNA sequences off the computer, creating a DNA code from scratch, and creating a nucleic acid chain is inserted into a blank cell. The DNA sequence takes over the cell, creating an entirely new organism. We also have the technology to place plasmids into other bacteria and have them take in the plasmid and portray the trait. For example, the common pea glow experiment performed by many AP biology students across the nation. The DNA sequence for this radiation-resistant bacteria has been recorded, and so if we combine the methane-producing bacteria with the radiation-resistant bacteria, we can create an organism that could convert the atmosphere on Mars to a hospitable environment, as well as survive and reproduce. The experiment for the Space Lab competition involves attempting to create the combined bacterium in a low-gravity environment, and seeing if it can survive as well as reproduce. This organism will be able to survive the cold, lack of oxygen, and the conditions on Mars. It will recreate the environment where liquid water can be sustained, as well as creating a pathway for new life forms. After the planet has been transformed, other organisms can be sent there to jumpstart the evolutionary processes seen on Earth.